Have you ever wondered how scientists study entire genomes? It's a fascinating process that involves multiple steps, each as intricate as the last. The exploration and mapping of a genome take place at many levels: species, individual, chromosome, gene, or nucleotide. The journey into genomic bioinformatics begins with the mapping and assembly of the numerous parts of each genome. Genome mapping, the first step, is the process of determining the relative position of all the genes on chromosomes in an organism's genome. To track genes, scientists use any detectable physical, behavioral, or chemical trait as a marker. The methods for determining these relative locations are varied, with linkage mapping methods identifying the relative order of genes along a chromosome and physical mapping methods determining the exact number of base pairs between specific genes. The next step is genome sequence assembly, a process that involves deducing and recording the exact order of every base and gene in a genome. To study an entire genome, scientists break up the genome into small fragments and clone all of the fragments. This collection of clones representing all of the genes in a given genome is referred to as a genetic library. Two types of genetic libraries are created: a genomic library and an expressed sequence tag, EST, library. Once the clones are assembled, they can be sequenced, sorted, and organized. Early methods involved sorting through libraries one gene at a time by repeated probing and deduction. More recently, a method called shotgun sequencing was developed. In this method, an entire genome is cut up randomly into segments of varying size. All resulting segments are cloned and sequenced. Then, by looking for overlapping parts, researchers put together the entire sequence like a puzzle. Finally, we have the age of automation. Robotic devices are now used to sequence a genome in a fraction of the time that it took to complete such a project only decades ago. These automated sequencing devices can quickly read many tiny sequence gels at one time. A laser beam scans each gel tube, and detectors identify each of the 4 kinds of tags. A computer then compiles the data into a string of letters. To summarize, genome studies involve a series of detailed steps: genome mapping, sequence assembly, the creation of genetic libraries, and the use of automated sequencing. Each step is vital in the process of studying and understanding the intricate workings of genomes. It's a testament to the incredible advancements in science and technology that we can now delve into the very essence of life on this microscopic level.